Hello, young anatomists. The purpose of this video is to take the notes that we'd write otherwise on how muscles work and control the information and reorganize it into a picture that's going to get everything we need to know here on one page. So let's organize this page a little bit by first folding our paper in half and then folding the paper in half again. And this will create 4 nice little sections, and then we're going to make another spot by folding this bottom part up a little bit. And here we go. This section down here will be reserved for some physiological plot part of our story. So we'll write physiology. And we are going to summarize 3 main ideas about muscle with our drawing. The first one that we'll do over here is I want to talk about how muscle is organized. Those notes are going to be mostly anatomy, where we're talking about names of structures. Along the top, we'll talk about muscle excitation. In other words, the interaction between neurons and muscles. And then finally, along the right side, we're going to talk about how muscle actually contracts. By discussing the contraction cycle. So let's jump right in with muscle organization. I want to start at the most gross anatomy level that we can, and that's by drawing, I'm going to draw an arm here. So there's our humerus, and we've got our radius and ulna as one picture here, and detailed drawing of carpals, metacarpals, and phalanges. And now I'm going to draw a muscle. So there's a muscle, and a muscle is considered an organ, and a muscle will attach to a bone with a structure called a tendon. Sometimes people confuse tendons and ligaments, and ligaments are structures that join bones with other bones. All right, so we'll label this guy as a muscle and remind ourselves that muscles are organs. And muscle, if we categorized it by tissue type, of course, it's going to be a muscle tissue, and a tendon is a connective tissue, and a ligament is a connective tissue as well. And what I want to do is, when I'm talking about organization, of course we want to know about this, but for this purpose, I'm going to zoom in on a cross-section of the muscle belly part. So let's zoom out on that. Let's zoom in on that, I should say. And we're going to make a nice little circle here for our muscle cross section. I'm going to leave a part open at the bottom. And let's draw some connective tissue covering of the muscle here. So this connective tissue covering is called the epimysium. And it's good that the epimysium has that name because it's on the outside of the muscle. So the epimysium covers the whole muscle. The epimysium is a connective tissue sheath or covering of the muscle. And all of these connective tissue coverings of the muscle are continuous with the tendon. So they join up with the tendon, and then the tendon joins up with the periosteum covering of the bone. So on our muscle cross section, we are going to draw some circles here. And we're going to draw one that's kind of coming out. So this one is pulled out. Now, each of these is a bundle. So muscles are kind of like bundles of bundles here. And there's connective tissue coverings that organize each of the bundles. So the epimysium covers the whole muscle, and the whole muscle is a bundle of these guys right here. And I'm gonna leave some space down here to pull one of these out as well. So this has connective tissue covering. One of these guys, this unit right here, is called a fascicle, and a fascicle is a bundle of muscle cells. And each fascicle has a connective tissue covering in here called a perimysium. Let's label it down here. So a perimysium covers fascicles. And a fascicle is made up of a whole bunch of smaller units, and those have connective tissue coverings as well. And that connective tissue covering is called the endomysium. So as much as it's confusing that all of these structures have similar names, their name kind of gives us a clue as to where they're positioned on the muscle. Epi, then peri, then endo. So the endomysium covers muscle fibers. So that makes this guy right here a muscle fiber. And if we're thinking about the organization of things, and a muscle is an organ, muscle fiber is a cell. And all of these things, if we classify them as tissues, they'd all be considered examples of muscle tissue, and all of these mysium coverings would be connective tissues. Now, another thing here, so we want to just kind of like organize this information. And on the outside of muscle, let's just put this information in list form over here. On the outside of muscle is epimysium. And then moving inside, we have perimysium. And moving inside of that, we have endomysium. Those are the connective tissue coverings, all continuous with each other, and then join up with the tendon at the end. The epimysium covers the whole muscle organ. The perimysium separates the muscle into bundles called fascicles. I want to make sure I'm spelling that right, fascicle. And then the endomysium covers muscle fibers. But we're not done just yet. Each muscle fiber itself is a bundle. So we could draw little dots in all of these. And then we can pull one of those out as well. And now this guy right here is called a myofibril. And myofibrils have two different flavors. So one flavor of myofibril is called a thick filament. and one is called a thin filament. And these are made up of proteins that we need to keep track of. So thick filaments are made up of proteins called myosin, and thin filaments are made up of protein called actin. So muscle is high in protein, and it's those proteins right there. So like as a food, meat is high in protein, and it's mostly actin and myosin. So I want to draw your attention to something. When we talked about cell organization, we talked about how cells have a plasma membrane, and muscle cells have a plasma membrane as well, and that's underneath the endomysium. So I want to just kind of like highlight that structure right here, and we'll just squeeze in the name, and that name for the plasma membrane or cell membrane of muscles is called a sarcolemma. So sarco and myo are muscle words. So this is the plasma membrane underneath the endomysium. So we could add this organization over here. So in our muscle fibers, we've got these myofibrils, and then the myofibrils themselves are made up of filaments called thick and thin filaments. All right, so now what I want to do, this is kind of how muscle is organized. To see how muscle gets excited to contract, I want to zoom in on just like this. We're going to take like this section right here, and we're gonna blow that up. So let's do that. And we're gonna draw a muscle fiber up here. Before we leave this picture, the excitation has to do with the interaction between nerves and muscles. So I want to draw a nerve fiber in here. So we're going to draw a nerve that comes up. Now keep in mind, this nerve would come from like the spinal cord and weave its way all along into the muscle. And it would, go in between these structures right here and kind of navigate in the connective tissue, and then make its way down onto a muscle fiber and put little things called axon terminal buttons onto the muscle. So we'll draw those in yellow, and that's going to help give us some perspective when we bring neurons into the mix up above. Okay, so let's draw our muscle fiber up here. So I'm going to draw one section with our muscle connective tissue covering on it. And remember what that connective tissue covering is called. And actually, let's draw it with that kind of like pulled away. So the endomysium is pulled off, and this layer will be our sarcolemma layer. All right, sarcolemma. And then we want to pull that membrane off and see what's underneath. And so let's draw the rest here. So here's the rest of our muscle fiber. Sarcolemma is the membrane of the muscle fiber. And we're going to draw a bunch of myofibrils in here. And we'll dot those up. So each one of these is a myofibril. And then myofibrils are even smaller, let's pull one of these out since we couldn't pull it out down there. Each one of these would be considered a filament. Okay, so we're a little bit organized here, and what I want to do is I'm going to draw these guys as going all the way back. All right, so we've got these long myofibrils here, and we're all set up. We just blew up a picture of this. And since we're talking excitation here, I want to add a neuron. So we're going to add one axon terminal here. And it kind of looks like a foot a little bit. And that's going on to nerve land over there. Now, neurons, they keep vesicles inside of their axons. So we're going to draw some vesicles inside of there, and those are called synaptic vesicles. And they store a chemical. They store chemicals called neurotransmitters. And when we're talking about the interaction between nerves and muscles, our main neurotransmitter is called acetylcholine, or you can abbreviate that as ACH for acetylcholine. And again, this is our neuron. And the great thing is we haven't had too many examples so far in our tissue of nerve tissue. So here's a good example right here. So what happens when it's time for a muscle to contract is these neurotransmitters, acetylcholine, are released onto the muscle. And the muscle has specialized structures to help receive that signal. Now, some of these structures are called T-tubules. So T-tubules make a network of kind of like holes in the sarcolemma. So I'll draw some of those and imagine these going into the muscle, and that's why we had to pull away the sarcolemma over here to see what's going on. So there would be like a hole in the sarcolemma, and then what would happen is the T-tubule would come down and like interact with each of these myofibrils down here. So let's label this guy. This guy's a T-tubule. And neurons, when neurons do their thing, neurons deal in electrochemical signals. And the job of a T-tubule is to spread that signal. So we're going to say that the T-tubule spreads the charge. So it spreads the signal of the neuron, and it gets that signal of the neuron from the outside of the muscle cell to the inside. And then the T-tubule will spread that charge to another structure that's on the muscle fiber, and that structure is like a network that's on each muscle fiber. So this structure in green is called the sarco. There's our muscle prefix again. Sarcoplasmic reticulum. Or you can call it the SR. And it sounds like the endoplasmic reticulum. But remember, the endoplasmic reticulum has the job of making proteins, and the sarcoplasmic reticulum has a different job. Sarcoplasmic reticulum stores calcium. So calcium isn't just for bones, calcium is for muscles as well. So now that we have all of these structures in place, let's talk about the physiology. Let's talk about how they work. So when a signal comes down a neuron for a muscle to contract, the neuron will release that acetylcholine into this gap called the synapse. And then the neurotransmitters will bind the muscle and turn this chemical signal into an electrical signal. And that signal starts to spread all over from the point where the neuron contacts the muscle. And that signal will spread, and then the T tubules kind of take that signal and spread the signal down into the muscle. So this yellow is now representing the electric signal from the neuron. And then the T tubules make sure that the signal spreads inside and gets to the sarcoplasmic reticulum. And there's so much T tubules in sarcoplasmic reticulum so that this charge can spread really, really rapidly. So then what happens is when this charge gets to the sarcoplasmic reticulum, all of this calcium that's stored in the sarcoplasmic reticulum rains down on these myofibrils and filaments. So I think it's important to get this down in words. So let's summarize that right up here. So the first thing in excitation that's going to happen is that the neuron is going to get a signal, and then the neuron is going to release acetylcholine. So the neuron releases acetylcholine to cause an electrical signal. Electric signal. Then our T tubules spread the electric signal to the sarcoplasmic reticulum. And then finally, the sarcoplasmic reticulum, when it gets that electrical signal, it rains down calcium onto the myofibrils and filaments. So this is the story of excitation, and this is our picture showing how that happens and the microscopic anatomy or subcellular anatomy that helps with that process. And then last of all, what I want to do is I want to zoom into some filaments and see what goes on with those filaments after that calcium rains down. Because remember, we're talking about muscle here, and muscle is a contractile tissue that can help with movement. So let's grab some of these filaments right here, and I'll meet you at the right side of our page. So filaments come in two different flavors, and those different flavors are thick filaments and thin filaments. So let's draw how those look up here. So I'm gonna draw all of my thick filaments in red. All right, so these are gonna be nice and thick. Let's label that thick filaments. And our thin filaments are gonna be interacting with those. Make these a little bit longer. We'll draw our thin filaments. So they kind of interlace with one another, like if your fingers do that. So let's label our thin filament. That are made of actin. And now I want to zoom in on some of the specialized structures of the proteins that make these up. So let's zoom in on one area right here of a thick filament. So thick filaments are made up of several of these proteins with kind of two heads on them. So we can draw a bunch of those with two heads. And they don't just go to one side, they go to both sides, and these heads are going to be the things that interact with the thin filaments. So let's keep this red for our label. So these proteins are called myosin. All of these are myosin proteins, and then this specialized part is called the head. So you've got all of these myosin heads, and myosin heads, if we then kind of like blow up one of these right here so we could see it a little bit better. All right, so we've got actually these two kind of long parts of myosin, and then they're myosin heads. And on myosin heads, at the very top here, it has an actin binding site. That's where the myosin thick filaments will bind the actin thin filaments. And then kind of in this area, there's an ATP binding site. So we need ATP to provide energy for muscle contraction, and that's because the ATP is going to interact with myosin. All right, let's zoom in on one of these thin filaments and see how that looks. All right, so we'll take one of those and we'll blow that up. And actin filaments, we can imagine those as looking kind of like a string of beads. And it's kind of doubled. All right, so actin filaments are these strings of beads, and we're going to put a little dot on each one of the beads. And that dot represents the area where myosin is going to want to bind. The head is going to want to bind right there. So this is an actin thin filament, and those have myosin binding sites, which are these dots right there. All right, so now I want to try to draw a couple of pictures to show how these interact with one another. So I'm going to draw in my actin filaments down here. As our beads on a string. And we're going to put those beautiful myosin binding sites on there. And let's draw some myosin heads that are going to want to bind those actin binding sites. And we'll take it easy with the myosin heads. We'll just draw two myosins with each having two heads. And here's those actin binding sites. And then what happens is that the myosin can't bind the actin binding sites because there's another protein in the way. So there's a protein that I'm going to draw on here in this green color. And it completely binds to and blocks all of these actin binding sites. This protein is called tropomyosin. And what keeps the tropomyosin in place is another protein that I'm going to draw in orange over here. And that protein is called troponin. So tropomyosin blocks myosin binding sites, and troponin holds tropomyosin. All right, so we've got 4 proteins involved here: actin, myosin, troponin, and tropomyosin. And what's going on in this picture, so this would be a muscle at rest. And what's going on here is that myosin can't bind actin because tropomyosin is blocking. And that's at rest. But then something changes when there's a contraction signal. When there's excitation. So let's draw that. And we're still going to draw our actin thin filaments. With the myosin binding sites on there. But in order to contract, the myosin has to be able to bind the actin. So we're going to draw our myosin down here. But when there's excitation, the myosin actually gets to bind to the actin. And the reason that happens is because now our tropomyosin that was being a pain in the butt before is moved out of the way. So here's what happens in this step. First, calcium release. By the sarcoplasmic reticulum. It causes troponin to move tropomyosin. And then second of all, the myosin head can now bind actin. So that's pretty awesome. We've got that interaction here. Another way to think about this is that troponin is kind of like activated to move by calcium. Then what happens is all of these myosin heads, when they grab onto actin, they change shape and pull on the actin, kind of like a rowing motion. That's called the power stroke. So at the end, we want to say that this is when the actual contraction happens. So we've got our actin thin filaments. We're going to draw in some myosin thick filaments here, but I'm going to draw something different about them. Now, instead of their heads pointing to the right, their heads are pointing to the left. And that's because these myosin heads have actually moved this way. And that's called the power stroke. To pull on that actin. So let's squeeze in right here what just happened. So myosin uses the energy of ATP to pull actin. And then what happens is this will repeat. So another ATP will come in and reset myosin, and then myosin will bind actin again and keep on doing this, what would be called a ratcheting effect of pulling the actin a little bit at a time. And that's called a contraction cycle. Contraction cycle. Now, I don't want you to forget that ATP is involved with this, so we'll put some ATP at the myosin head there, ATP at the myosin head over here, and then we'll say that energy is used for the power stroke. So those are our three stories. First, we talked about muscle organization into bundles of bundles. And then we zoomed in to talk about how muscles can be excited by neurons for contraction. That excitation involves an electrical signal, which eventually leads to calcium release by the sarcoplasmic reticulum inside of the muscle cell. Finally, once the calcium rains down on these myofilaments, that causes troponin to pull tropomyosin out of the way, so that myosin heads can grab onto actin filaments. Myosins can change shape and pull on the actin. Now, all we have left here is some physiology. And this part's going to go pretty quick. The main function of muscle is to produce movement. So again, we've got we'll draw our arm here. And I want to draw very deliberately our tendons so we remember that anatomy. We'll draw very deliberately here our muscles. And so what happens is the muscle will contract and pull on a bone to cause this movement right here. So contract, and then another important thing that muscle needs to be able to do is that muscle needs to be able to relax as well. And that's why we need these bundles of bundles and this kind of like hyper organization right here, is that muscles have to keep contracting and relaxing over and over again. So if we drew a contracted limb over here, now that bone would move. Here's our tendons, and that muscle belly gets like really big. Now what happens is you can actually see contraction at a microscopic level. So I'm going to draw some of our thick filaments in there, and notice that the thick filaments look the same when the muscle is contracted or relaxed. Now I'll draw in these thin filaments. If I can draw today. And when these thick filaments pull on the thin filaments, how we get the muscle shortening is that now the thin filaments are pulled all the way in to kind of like this midline right here. But the length of the thick filaments and thin filaments doesn't change. So here's our contraction. I'm going to use orange to help us remember that calcium is involved in this process right here. And the fact that these thick and thin filaments slide over one another is called the sliding filament theory for muscle contraction. All right, so that's physiological function number 1. Skeletal muscle produces movement. Number 2, muscle is really important for maintaining posture. That's how you can sit up in your chair right now or stand up and not fall over. Muscle stabilizes joints. Sure, there are ligaments in there to hold these bones together, but having all of those muscle structures in place joined to the bones as well keeps those joints from disarticulating or kind of popping out of the socket or becoming dislocated. And then last of all, skeletal muscle really helps with temperature homeostasis. And we've talked about this already in kind of like multiple forms here, but I just wanted to recap whenever we can. So let me use a different color. I'll use purple here. So cellular respiration normally makes heat. And your muscles, when they contract, we talked about how your muscle uses up ATP. So that means to make more ATP, your muscles are doing a lot of cell respiration, giving off heat as a byproduct. So you're constantly giving off heat, whether you like it or not. When you're too cold, your muscles contract extra, and that's shivering. So shivering increases the amount of heat that muscle will give off to try to increase your body temperature. And then we talked about some other muscles called erector pili muscles. And those are the muscles that are attached to your hairs. So when erector pili muscles contract, they raise the hairs to trap heat against the body. So when you're too cold, you can shiver to make more heat, and you can help trap some of that heat from leaving your body too much. So here's our muscle physiology. Here are some stories of anatomy and physiology. Now we control this information, and it's nice and summarized on this one page for us.